Hello everyone. Today we have a very special guest with us, Professor Kesavardhana Sannula, who is an assistant professor at IISc. His interests include a wide range of topics, but mostly is interested in RNA and also viral host interactions. So from his lab, there is a recent study which is going to focus on H5N1 virus and its implication on how it can spread in the human coming from the non-human host or other mammals. So this study is very exciting because it is very important for not just individuals, but also for officials who are making the policies at higher level. And we'll talk with him and try to understand what was the thought process that went behind this study and what are the outcomes from this study that can have some impact on the future. So thank you, Professor, for joining the talk today and giving your time.
Yeah, thanks, Anjani, for having this discussion. Appreciate it.
So, Professor, I would like to ask you a very basic questions. So your interest, as I went through your web page and your research articles, I got to know that you are interested in viruses and RNA. So I would like to ask what brought you to this field? Like, why were you interested in immunology or inflammation biology? So what was your inspiration behind starting like the seed that took you to this form now?
Okay. Yeah, I think I agree with you what we are working on, like viruses and how these RNA viruses particularly, how they communicate with our immune system and how does it work. So typically, if you look at viruses, right, their genome is much smaller than, bacteria or whatever you can say. They're much smaller genomes, and especially RNA viruses even have much smaller genome. Let's say if a DNA has 100 kb genome or sometimes more than that, RNA viruses mostly have like 10 kb, 20 kb maximum, or sometimes it's even less. Okay, so that's fascinating having such small genome, but they turned out to be much more pathogenic than the rest of the DNA viruses, right? So that means they have evolved so much that even with very limited number of proteins or genome, they could able to handle the entire host system, and they can happily propagate in the host. I mean, that's something amazing that people are still trying to understand. The other thing is, so when I started my lab, our goal is to look at why these viruses, particularly pathogenic RNA viruses, they're happy in bats and birds, right? But when they come to human beings, they're causing all the chaos, right? All the disease, particularly these genetic diseases like SARS-CoV-2 infection, bird flu, and Zika virus infection. There are multiple of them, even Nipah. Why? We asked a simple question: is that why these hosts like bats and birds could able to handle these viruses? Why not we? Right? And understanding, I know it's a very broad question, but understanding this will give us a right mechanisms to target in case if we come across with any pandemic in the future. That's one point. And we also want to study that can we come up with certain aspects where can we predict the pathogenic or pandemic potential of a virus, which is there in the bats and birds? Because now a lot of people are sequencing viruses which are there in multiple of reservoir hosts like bats and birds. Just by looking at the genome sequences, can you tell that if this particular virus comes to human beings, it can become like SARS-CoV-2 or like avian influenza, right? But that will be the ultimate thing. Then it's when we call it as in terms like pandemic preparedness. That means you are prepared for the next pandemic before even it has come to you. So that will come with a lot of knowledge that we need to have. So that's why we started understanding what's the difference between different hosts of handling these viruses. So in this particular way, we published some of our studies where we identified that bats especially, they have very interesting mechanisms of controlling cell death and inflammation compared to the human beings. So they adapted in such a way that virus can nicely replicate in them and then exist in them, but they don't cause much of a damage like cellular damage or inflammation. But if it comes to the human being, that cellular damage inflammation is very high. And not only that, these bats and birds, they slowly help the virus to acquire pathogenic traits. So when they come to the human beings, they readily act on human beings. And then other one, the one which you are going to ask me about avian influenza which is known as H5N1, which is very well known like bird flu. We became interested in that let's say if birds have tons of viruses, let's say many viruses, why only this particular H5N1 became panzootic or pandemic? Why not all the strains? So that's a simple fundamental question one need to ask, because then you can define that why this particular virus has become pandemic virus because of particular features or molecular characteristics. Then you can say that if using that information, you can always predict the pathogenic potential of new viruses. So this is where right now we are working on.
That's a great answer. Actually, my question was going to be about why did you choose this particular clade of virus? And you already answered it because of the zoonotic potential that is seen in the molecular features.
Correct.
So,
maybe I'll add one more line to that. So this particular clade called 2344B, okay, it's a H5N1. So there is a lineage called Guangdong lineage because it named as Guangdong because the first virus H5N1, which was like sequenced and characterized, is from Guangdong place in the China. Okay, and the lineage started like that. The current Guangdong lineage virus is 2344B, H5N1. If you look at birds, there are variety of avian influenza viruses, not only H5N1, like H5N8, N6, H9N2, there are multiple. Okay, but suddenly in the recent time, this H5N1 again emerged. People thought that it's endemic, it may not come back, but suddenly this particular virus called 2344B has emerged. Then that's where we became interested. Why this suddenly emerged? We thought that it's gone. Right? So yeah.
Yeah, it's very interesting. You talked about different reservoirs for viruses. So it's not just that there are birds, but also there are some cattle and all the other like pigs and all the other bats.
Exactly.
For example, for SARS-CoV-2, that was one of the reservoirs.
Yeah.
So what is the role of a reservoir in like spillovers in the humans? Like, are there some specific genera that are more susceptible to these kind of viruses, RNA viruses that you particularly study, or is it like more evenly spread across different organisms?
Yeah, right now we are focusing on the viruses which can potentially infect humans and can replicate in humans, because that's the biggest problem for us, right? For example, what we learned from COVID is that so this virus could be able to nicely transfer to human beings and replicate and spread all over the human beings. So the viruses which have that potential are major concern for us, especially because of the economic and public health burden that we are going to face if they come to human beings. Right? But at the same time, we also are interested in viruses which are very well adapted in the reservoir host, but not well adapted in the human beings. For example, avian influenza, H5N1. It nicely replicates and propagates very well in birds, wild birds, and poultry. Okay, but for human beings, it still cannot make a regular upper respiratory tract infection because they don't have a receptor there. But if you somehow inhale and goes to the lower respiratory tract, it'll be very chaos. It kills all the, it creates a respiratory problem and then damage and you get the disease. What does it mean is that this virus can jump to the human beings, but it is not adapted yet to transfer among human beings. So that means the person who is infected may not be yet able to transfer the virus to the other people. But the problem with 2344B that right now we are working, it has crossed barriers that it is infected many mammal species and it is adapting in the mammals, which is a concern for human adaptation.
Okay, so in simple words, it's not just the infectious rate of the virus, but also how well it is able to transmit across the same species. So for example,
Correct.
if like you give the example, if the virus goes to your lower respiratory tract, but it is not going to transmit to other human beings, then it is not so much risky. But if it is
Correct.
going to transmit to other human beings, then that becomes higher risk.
Exactly.
So as far as I'm able to understand, this particular clade that you are studying, this also has higher transmittability, or is it still in the study phase?
In the human beings, no, it hasn't acquired the ability to transmit among human beings, but it is transmitting among other non-human mammalian species. Like people have reported that cattle are dairy cattle or cows, and in some of the sea lions and minks, people have reported that they are transmitting among mammals. which is a concern because then it's slowly adapting to the human system, or especially, mammalian system, where human beings also one kind of mammals.
Yeah. So it's basically getting closer to humans as it is adapting, like across the evolutionary tree, it is coming closer to humans
Exactly.
from cattle and yeah. Okay, got it.
Exactly.
So these changes in the virus happens over like some short period of time because these are RNA viruses and RNA is quite unstable as compared to DNA. So one would expect that the rate of mutation would be higher in the RNA viruses. Plus, you started with the information that the genome size of these viruses are quite small. So, what mutation rates are like found in this virus that can help in the progression of this adaptations? Like,
Okay.
is it higher as compared to other clades, or is it comparable, but still it is making changes in the important regions that is giving it the advantage?
Exactly. So the virus, the way it mutates, especially RNA viruses, for example, in this case, influenza, the way it mutates, it depends on which kind of host it is in. For example, if it is in avian influenza, when it is in birds, it naturally always acquire mutation, but the mutation rate may be low because they don't have any evolutionary pressure from the host. They're happy living there. But when they jump to the new host, in order for them to sustain in the new host, let's say it has jumped from birds to cows. Okay, cows is kind of a new system for them. They can replicate, but in order for them to efficiently replicate, they have to rapidly evolve. Okay, that is called adaptation or adaptive mutations. If they adapt or if they acquire the mutation that can adapt, they will propagate. Otherwise, they will be they will not able to sustain in that particular host. And the second thing is naturally RNA viruses, because of their small genome, the rate of acquiring mutations or rate of acquiring adaptations is a bit much faster than the DNA viruses. And not only that, most of these RNA viruses have error-prone polymerases, and they tend to naturally induce random mutations, and some of them will be selected if they are useful. If they are deleterious for the virus, if they don't confer any fitness for the virus, they will not be selected. So the host will definitely have a bigger impact, how fast they can evolve or how fast they can adapt.
So another question is that all these studies that are performed for calculating the transmittability rate or the infectious rate, are these like the study from your lab, the recent one? Are these more like bioinformatical studies, or were these performed in the experimental system in your like some cell system, and then based on that you calculate the chances of spreading in humans?
Yeah. So right now, the big problem with avian influenza or highly pathogenic influenza viruses is that if you have heard a term called gain of function, so that means you are not allowed to mutate the viruses experimentally to study them. So it's a limitation for influenza virus biologists that you cannot introduce mutations that are appearing new to study whether how are they acting. So the characterization will become limited. So that's why what we took an alternate approach for this particular study is that first we track the mutations in each mammal. Like let's say if you take a virus from influenza, which is from the birds and that is transmitted to the cows, once it infects cows, how is it propagating with the generations? Is it acquiring mutations? Is it not acquiring the mutations? If it is acquiring the mutations, are these mutations matched to the amino acid residues which are there in human influenza viruses? So that indicates that, look, these viruses are gaining mutations that can be suitable for human influenza infection. That's one part. The other part is there is huge literature available where many scientists have characterized what mutations required for an avian influenza virus to turn into human influenza virus. So that means what are the critical mutations that H5N1 or avian influenza acquire so that it can happily replicate or transmit among humans. So that is very well characterized in the cell lines. Now we using that mutations as a reference for us. Then we start comparing the mutations that the new clade is acquiring in the different mammals like cows, sea lions, foxes, seals, bears. Then we'll track them. Then once we have them, then there are two types of categories you can make it. For example, whether the mutation that is acquired in that particular virus, is it required for that only that particular mammal, let's say cows, or that mutation can in principle be useful for any other mammal. Okay. So for example, if a mutation that we have identified is only there in cow viruses, okay, but not there in human viruses, and then it cannot be useful for human viruses, we call them as a barrier mutations. That means the avian influenza virus, which has come to the cows, acquired a new mutation that is very mammalian specific or cow specific, that may not be useful for human adaptation. But there are other mutations, we call them human adaptive mutations, which matches to the human viruses and then can bring certain adaptation to become human adaptation. So now based on these two mutations, two categories, we come up with a simple mathematical model. Okay, now we have viruses which is nicely circulating in diverse mammals. If you compare all these viruses from diverse mammals, which one has more human adaptive potential than other mammalian viruses? So then we establish a simple mathematical model to come up with this kind of numbers where we can have certain idea about how well these viruses adapting to the humans in these particular mammalian hosts. Hope I made sense to you, but you can ask me if you didn't get any point.
Yeah, it was very interesting. So as much as I'm able to understand, I think this whole idea of identifying the viruses that have higher human adaptive potential, this would have come first, and then you would have identified the clade, which is more, or was it already based on some literature that you identified this particular clade and then performed analysis across different organisms? So let's say you have H5N1 and you want to identify among 10 of its strains, which one is more human adaptive potential. So then you start the analysis and then you identify which of them is the lead, and then you do further analysis for that. Or was it based on some literature already?
Yeah, right now the current study is based on the literature, but now there are other groups which are actively testing how these viruses from different mammals is nicely replicating in very like a mammalian model called ferret model. It's instead of mice for influenza, people use ferret because they are very good for transmission studies. So now they're testing them to see that which one is more transmissible among mammals, especially like human-like mammals.
Got it. And so all this data that you source for our analysis, this should come from some like global repository, or is it like done with some collaborative project from your group and some other groups?
No. Right now, I think the influenza community is so open and very active, especially because of this 2, 3, 4, 4-week lab. They're depositing all these things into a global repository called GSAID, GISAID. It's reported in the paper. It's like they have very well documented data of the moment they get a sequence, they deposit it and they will categorize them, which type of influenza virus is there, which clade virus is there, is it avian influenza or human influenza or cattle influenza. So very well characterized. So basically you have n number of sequences available, but only thing, the challenging thing we had when we were working with is that the mammalian infections just started from 2022 onwards. Okay, so the number of sequences deposited for each mammal is limited. For example, let's say for cows, sea lions, and foxes, we could get bunch of sequences so that we can analyze and come with some statistical numbers, but not for all mammals. Okay, so surveillance also it's important, but whatever people have been like doing the surveillance and sequencing, they are actually depositing the information in GSAID. And that's where we tend to get the sequences. But rest of other things is completely we develop the pipelines using computational models to analyze each set of, to starting from, there will be a lot of mutations. You need to have a screen that are there useful mutations or not useful mutations, or missense mutations or nonsense mutations. Then you take that to the next level.
That's actually great. Like until and unless we collaborate, it's very difficult to tackle these challenges. So
Exactly.
it's great to hear that there is
These
a
kind of
community.
Yeah, especially these kind of things, you definitely need to have collaboration or understanding among people so that the new information that anybody analyze or understand will be disseminated faster.
So now that we're talking about more on the human aspect of this, would you also like to give some suggestions, like once we identify that this particular clade is more having a higher human adaptive potential, should like the officials be concerned about this, or is it slowly mutating? It is far away from the tipping point, or is it near the tipping point? So the higher officials should be concerned about this or not at this point.
Yeah, definitely. Like, I mean, I would say that, one need to be very concerned of, especially the healthcare system, because if you look in US, Canada, and I think even in UK, they understood the problem of H5N1, especially this current clade, and they do anticipate that. I mean, at least the community anticipate that this H5N1 could be the next pandemic. So that's why these countries now they are prepared in such a way that they're manufacturing already vaccines in order to be ready for the pandemic if any kind of outbreaks happen. The thing is, right now we are still lucky that there is no human to human transmission, but people have seen that the viruses which are there in cows and some in sea lions, they acquiring human mutations, and also cow viruses have the ability to bind the receptors which could be there in upper respiratory tract. So that's a concern. And not only that, this particular H5N1, it's not only entering through the respiratory droplets, it's also communicating through other means. Like in case of cows, the major transmission is through milk or mammary glands. And it's actually causing, in other mammals, it is causing many neurological issues. So still we don't know what kind of pathological condition this virus could be there. But definitely there's something that we need to have certain preparedness. For example, let's say in India, maybe we should have more genomes of avian influenza virus sequenced and see that what are all the different 2, 3, 4, 5B viruses we have, like what kind of mutations we have. Then based on that, if we could come up with certain formulation of avian influenza vaccine, that would be better thing. But we, as our lab, we are also in the process of actually would like to identify therapeutic molecules which could specific this 2, 3, 4, 4B H5N1, and then test them in laboratory and then see that if we can come up with something which is more efficient.
That's great. Actually, you are not just identifying the problem; you are also trying to identify the solution. So
yes,
that's very interesting to hear. Yeah.
That's the ultimate goal for any of us who are working in this angle.
So one more thing I wanted to ask from a general audience point of view, that should they take some precaution? Is there a way that they can shield themselves from this maybe in future pandemic, or should we just hope that there will be no pandemic and will be secure from this? So, or they can take some steps on a personal level to shield them from this.
Yeah, I think definitely is something that it's still in our control, so we should do this. So for example, if you look at CDC, Center for Disease Control, they are very well documented now that most of the human sporadic infections which have happened recently with this particular variant is in the people who are working in dairy cattle or people working in the poultry. So that means the human contact close to these kind of host systems which have these viruses is causing the human infections. So having a precautionary note of not going close to them or handling them without any precautions may not be a good idea. If you know that, let's say this particular area or the flock of birds or cows or any other animals, like even in the cats, if they have viruses, I think it's better that we don't too indulge into touching or getting the respiratory droplets into us.
Yeah, so I think one of the steps that people can take is they should diagnose whatever their cattle or poultry are having. If they are having some symptoms of influenza, they should
just do the testing. There are very conventional tests are available.
Yeah. instead of waiting longer and then allowing it to spread in humans, which will increase the
Exactly.
risk.
And even NIV, if you know National Institute of Virology, they're very well, onto it right now. They started, there are a couple of publications recently where they try to publish 2, 3, 4 4V sequences from India. And there is one more institute, Public Health Institute, in one health institute in Bhopal. They also reported one cat sequence, which is good that now people have started sequencing viruses in Indian settings, so that you know that these kind of animals might have these viruses.
So I would like to ask you a last question about your study. So every study that we do as a scientist, it's very close to our heart, right? And I believe this would also be very close to your heart, and you had some hypothesis to start with. You would have set with some objective and expectations. So were there some surprising things that came out of this study that you didn't expect at the beginning, and then you were surprised at the end that this was something very new, or like a eureka moment that you found during the study?
Yeah, definitely there is surprising thing, because multiple of them. But one thing I'll tell, when we were trying to analyze all the mammalian sequences and we were making the mathematical model and everything, we were expecting that cattle viruses turns out to be more have like more close to the human adaptive potential. But no, it's not cattle. These fox viruses have more human adaptive potential. But there are very less number of foxes got infected, but still the virus which is already there in them have this, which is surprising. Maybe it's that their system has given evolutionary pressure on virus that they acquired these things. That's very surprising for us, which we didn't expect, but we should go by data, or we should go by the analysis which we identified. And the other thing is, I'm sure you must be know that many people are working, especially Western countries, and then be it because of the resources or the data or the expertise they have, they're working. But one thing what we realized before we started is that everybody is working in a conventional way, as that, oh, now there is a mammalian, the virus has infected mammalian, isolate them and then look at cell lines, what is happening, is it more pathogenic than avian influenza or not? But our lab is a bit distinct from others because we have an expertise in understanding a problem from biophysical to biochemical to cellular to organismal level. We have a different set of advantages of where we can try to understand even at molecular level and try to interpret that at cellular level and see what's happening. That's because of our, especially my expertise or my PhD in biophysics from IISc itself, where I had a lot of experience in understanding structures of proteins and mutations or variations, how the virus will evolve. So that helped us to look at multiple layers. Then we started developing, can we develop certain small mathematical formula or model to just see that what are the numbers can tell us. So that is feasible, where that is a bit feasible for us than many other groups is because of our prior knowledge or expertise in these areas.
That's really interesting, the way you integrate different fields of biology and your expertise from the biophysics, and then the current work that you're doing. It's very interesting because in the end, it's all the amalgamation of the knowledge that we have gained over the years and the experiences that we have gained over the years that leads to the future discoveries. So it's very interesting to hear from you from such a diverse perspective and all the different set of tools that you have currently. And finally, before closing, I would like to get your message for the audience. Most of them are young generation, so they would like to pursue maybe research in future. So probably you can say some inspiring words for them so that they can also do some exciting work like you are doing currently and pursue their dreams in the field of research. So yeah, whatever you would like to share.
Definitely, I'll definitely share what's my experience so far, although I have a limited experience. But thanks, Anjane, for asking the right questions, because that's important for the people to know. I would say for the people who are aspiring their career in science or something, or even in any career in that matter. So in science, things goes by, are you curious about a question? So do you have a question that makes you curious? Then that automatically drives you to work on it. And then problems or difficulties are common for everybody. Everybody will have. But generally, when we get problems, we think that, oh, we are in problems, we have this problem, or we have these difficulties. But in general, this is there for most of the people. The one who sustains, who is patient, who is willing to work to make some change, will definitely move forward and find something. I mean, this is what I experienced personally from my side with limited experience. But I think if you ask many people, that's what they say.
Thank you, Professor. Thank you for your time. It was great talking to you and knowing about your current study. I hope you
Thank
do
you very
many
much.
more works in the future and help us find a cure for the virus, and also do some exciting work that can help us find cure to many more diseases. Like your PhD students will go on to set up their labs, and they will also work in some cutting edge field that will help protect the lives of so many people and change it. So thank you for doing this great work and
Thank you.
a service for the society in true sense. Thank you very much.
Yes, it's my duty. Yeah, I'll definitely do. I'll continue to do so. Thank you.
Thank you, Professor. Thank you.